Swayam Prabha. Digital India. Educated India.
So, hello everyone. So, today we will discuss about the biocompatibility of biomaterials. So, we will quickly review the last lecture. So, in the last lecture we discussed about the cell adhesion mechanism, which are mediated through different proteins, including the cell receptor proteins, ECM proteins, then also some pre-adsorbed protein. We discussed cell adhesion molecules which participate in the cell and cell surface interaction. So, we discussed there are 5 different classes: integrins, catherins, selectins, immunoglobulin and mucin. So, we discussed one example like integrins. So, they are 24 heterodimeric structure. So, integrin units attaches with the cell through the cell membrane, and its one end attached with the cell adhesive protein, let us say fibronectin, which is the part of extracellular matrix. Now, this fibronectin, one side it has attached to the integrin and other side it will attach to the surface. So, that is why the cell surface interaction take place, and it is mediated by different proteins. So, in many times in absence of protein, cells secrete the protein to facilitate the cell attachment, and in the absence of anchorage or attachment, the cell death takes place. You can say the cell apoptosis. So, followed by that we discuss, as there is an interaction of cell with the surface, so there we discuss different intermolecular forces, and also we saw the DLBO theory, how it includes both attractive and repulsive part, which is applicable to the cell surface interaction. Then we calculated the work of adhesion, particularly in terms of Gibbs free energy. So, we discuss the spreading coefficient should be less than 0 in order to facilitate the spontaneous attachment of the cell, and that work of adhesion can be tuned by tuning the surface energy of the solid surface. Then we discuss how we can analyze the kinetics of cell adhesion. So, we discuss the cells coming from the bulk to the interface, then from interface to the surface, then from bulk to interface it is having a mass transfer coefficient, and from that interface to surface it is having the adhesion rate and detachment rate or resorption rate. So, based on that we ignore the mass transfer part and particularly the death rate during the initial cell adhesion, and we derive that we can fit the experimental data either in terms of number of cells or the surface coverage with time to determine the rate of cell adhesion. So, in today's lecture we will discuss further how we define the biocompatibility of the biomaterials, what are the different factors which play the key role during defining or assessing the biocompatibility of biomaterials. Then we will discuss in vivo methods as well as the in vitro methods to assess the biocompatibility of biomaterials, and finally we will see if you want to develop a new biomaterial, what should be its characteristic features. So, biocompatibility, as the name suggests, it should be compatible with the body fluid or the biological environment, So, it is defined as the ability of a material or ability of a biomaterial to perform the desired function, that too in the harmony of host cells. Means it should work or it should synchronize with the host cell when a biomaterial comes into contact or when a biomaterial is implanted inside the body. In other words, the same thing can be said as that the ability of a biomaterial or a material to perform an appropriate host response in a specific application. So, now with this definition it comes, you cannot have a common definition of biocompatibility because it depends on a particular application, but the common thing is the appropriate host response. So, any material it should perform and its intended function with appropriate host response. Now, what is the intended function? That will depend what is the desired application or what is the specific application. So, basically depending on the application we will define what should be the criteria for biocompatibility or what should be the criteria for the application of biomaterial as implant or other application. It depends on the biocompatibility. So, you can say it is either way. If it is biocompatible, then biocompatible for which application? Because depending on the different application there will be the different requirement that we will discuss in the next slide. So, as we discussed during the cell surface interaction, whenever there is a implantation of a new biomaterial, so there will be the inflammatory responses depending on the complement pathways, the classical pathways or alternative pathways or lectin pathways. So, the severity and the duration of inflammatory responses, this indicates the biocompatibility, or you can say this is the indicator of the biocompatibility of the biomaterial. So, what is expected from a biomaterial is it does not induce acute or chronic inflammatory responses, and also it does not prevent a proper differentiation of the cells which surrounds the implant, or you can say the proper differentiation of the tissues which surrounds the implant. So, we can say if material is biocompatible, it does not induce acute or chronic inflammatory responses, the first thing. Second thing, it does not prevent the proper growth of the tissue or the cells. And again, as we discussed, this proper growth or differentiation, it is subject to the a particular application, that what is the intended application of that particular biomaterial. So, as we know this cell surface interaction, so as we do not have any control over the cells. So, if you want to tune the cell surface interaction, so it can be tuned by the changing the property of material, because the property of material determine or regulate the cell surface interactions. and which determine the response of host cells or the host tissue, and this biocompatibility can be relative to the what is the type of cell and what is the duration. And finally, that what the immunoresponse and the repair mechanism. So, these are the two important things, immunoresponse and the repair mechanism of the body govern the biocompatibility. So, the biocompatibility depends on the property of the material. Again, where it is applied, the which type of tissue or cells are exposed to the biomaterial, what is the duration of application, whether it is a implant, it is a drug delivery, it is a sensor, it is a scaffold, and in all the cases the immune response as well as the repair mechanism of the body govern the biocompatibility. So, as I told, there can be a number of application of biomaterial. So, here I have listed few important classes of materials, or you can say the biomaterials, starting from the metals. So, metals mostly used for the implants and orthopedics like dental implant, bone implant. So, we mostly use the titanium and titanium alloys, stainless steel. So, it is having a different application. So, if it is being used for the bone implant, its mechanical property, its other characteristics should match with the bone. Now, different polymers are also used. So, they are used as different body parts, different joints, or even the sutures. So, these are the application of the polymers. So, what property we expect from the implant, the same property we cannot expect from this, particularly just say the sutures. So, what happens, let us compare these two cases. So, we should have the different mechanical property. It should have different mechanical property as well as the duration of this, particularly the sutures and the implant is different. So, duration is different. Their environment is different, application environment is different. So, depending on these factors, we have to define the biocompatibility criteria for the implant will be different, and biocompatibility criteria for the sutures will be different. Apart from the polymer and metals, the hydrogels are also used, particularly for contact lenses, for tandem materials, as a bond healing bioadhesives and cartilage. Bioresorbable materials which degrades with time. So, they are also used for the sutures, for drug delivery, and in case of orthopedic implant they are being used for the fixation devices, because when they are used as a screw or fixation, so with time they should automatically get degraded or dissolved. Same is true for the drug delivery also. In case of drug delivery, there should be sustainable release of the drug, at the same time the sustainable degradation of the material should also take place parallel or simultaneously with the release of the drug. Apart from this, the ceramics and glasses are also used, particularly for eyeglasses, tissue culture, plate, tissue culture, flask. This in dentistry, especially for the filling of the dental, we use various ceramics like hydroxyapatite and some other its combinations. Some natural polymers are natural materials like the proteins, the silk protein, collagen, some natural polymers, natural carbohydrates. They are also used as a matrix or support for the preparation of, let us say, scaffold preparation, for the support, particularly if you just consider the silk. The silk is a protein which can be mixed with other polymers or hydrogels to improve its biocompatible properties or improve its biocompatibility. Apart from this individual, we can also make the composites. So, particularly let us consider the polymer composites. So, polymer composites, polymer can be reinforced with the fibers or it can be reinforced with the some particles. So, in that way we can improve its mechanical, physical as well as the chemical property of those composites. So, few example of the composites for the fracture fixation and this joint replacement like knee joint replacement and so on. So, now if you consider these are the different classes of materials or biomaterials. So, we cannot have common criteria. Of course, there will be some criteria that common criteria is no immune response as well as the proper repair mechanism, but the other criteria it will differ from the application to application or even it vary from the material to material. So, how do we assess the biocompatibility? So, we go for both in vitro as well as the in vivo analysis. So, we start with the in vitro analysis. So, in vitro approach estimate the compatibility of material with specific cell types or specific cell line, and it does not involve immunological component similar to body, because we are performing in vitro. One example of in vitro is the cytotoxicity study. So, when we perform the in vitro analysis, what it does? It minimizes the number of experiment required for the in vivo analysis. So, you can say it minimizes the animal or you can say minimizes the number of animal studies So, based on the in vitro experiment, we choose which one is the best material which need to be tested inside a particular animal model. So, that animal model may be the rat, it may be the rabbit. So, after in vitro approach, we go for the in vivo approach, which gives the actual effect of biomaterial implant inside the body. Here we have with respect to its immune system, because here we are dealing directly with the living system. So, there will be the immune response also. And for testing these two techniques, there are various protocols, that ASTM and ISO protocols. So, which are applied or which are followed for testing these in vitro or in vivo assessments. So, in case of in vitro assessment, we test the toxicity, we test the leachability, and we test the hemocompatibility. The toxicity means some toxic effects. So, toxic effect may be the death or the killing of cells, or you can say the some alteration of the membrane permeability or some inhibition of the enzymes at the cellular level. Leachability means once a biomaterial is implanted, it releases some metal ions or some its chemical constituents, chemical compounds which may be toxic. So, what you have to do? You have to keep them in some simulated medium. So, we use the simulated fluid. And with time analyze what is the components and as well as the what concentration of extracts or you can say the leachates. So, in this way we analyze the leachability, and the next is the hemocompatibility, or you can say the blood material, or you can say the blood leachate interaction. So, you can say this is like a blood material interactions. So, it may include the clotting study, it may include the adsorption, because blood contains a lot of serum proteins. Also, it may include the platelet adhesion. So, depending on this blood material interactions, we comment on the compatibility, or you can say the biocompatibility of the material. So, it is desired that it should give the favorable interactions with the material. It should not leach out the toxic compounds, as well as it should have no or cytotoxic effect, or you can say it should be cytocompatible. So, for cytocompatibility we apply, or you can say the cytotoxicity, we apply three different methods: direct contact, agar diffusion, or the MEM elution. So, in case of direct contact, we grow the cells, then we put the material directly into contact the soil for certain time, then we analyze the cells, how many cells are dead, life, and other its biochemical features. Similarly, in case of agar diffusion, we grow the cell, put the agar to solidify, then we put the material over the agar or on the agar, and we incubate for certain time to see how the cells behaves, whether cells are surviving or they are dead due to the presence of the foreign material. And similar to that, in case of elution, we first elute the leachate or extracts in the media, then we grow the cells and put that media with that extracts or leachates, and then we incubate for certain time interval to analyze the behavior of the cells. So, as we discussed, in case of direct contact, first you grow the cells in the petri dish or petri plate, then you place the samples directly on the cell, incubate for let us say 24 hour, 37 degree centigrade in a CO2 incubator with a controlled humidity and CO2 level. Then after that you fix the cells and analyze using different staining for the live and dead cells. So, this is a direct contact method. Agar diffusion method, it is very similar to the direct contact method, only difference is after the growing cell on the plate, we pour the agar over the cells and wait till it is solidified, and once it is solidified, then we place the samples, we incubate it, and we follow the similar procedure as we discussed for the direct contact. Now, in case of elution method, we are having first step is we have to extract in the serum free media, or we can also use 0.9% saline. Then you have to separately grow the cells in the petri dish or petri plate. Then after that you have to add the extract to that cells, and we have to incubate here for a bit longer time, that is about 48 hours. Then you have to fix and analyze the live and dead cells. So, these are the three methods, that direct contact, agar diffusion or the elution method. So, once we got the exposed cells or incubated cells, so one method is the MTT assay to analyze the cytocompatibility or cytotoxicity. So, if a cell is metabolically active, it produces NADPH, which is depend on the mitochondrial dehydrogenases for controlling the electron flux. Okay. So, what it says, if it is a active cell, then it will have presence of dehydrogenases. Now, what happens, MTT is a dye which have this diphenyl tetrazolium bromide group, and the presence of dehydrogenases, it reduces it to form the pharmajan. So, this tetrazolium converted to the, or you can say reduced to the pharmajan, and now this initially this MTT was yellow color, and after the reduction it becomes purple color. So, here you can see in the healthy cells or live cells, it will have this succinate dehydrogenase, and the succinate dehydrogenase particularly works, or you can say reduces the MTT to the pharmajan crystals. So, the yellow color converts to the purple color, and this color intensity, or you can say whatever this pharmajan crystal is formed, we can again solubilize them in another solvent, particularly we use the DMSO or isopropanol, and then we measure its intensity using a spectroscopy methods. So, what happens, the amount of the pharmajan crystal form is directly proportional to the cell viability. So, how we incubate cell with the biomaterials, then after certain time intervals, 1 day, 3 day, 5 days, 7 days, we add the MTT, then we incubate for 4 hours for its reduction, or you can say the formation of pharmajan crystals. Then we dissolve the pharmajan in some solvent like DMSO, and then we measure the absorbance using a multiplate reader. As it is a purple color, we use the 570 nanometer. So, the intensity of the purple colored pharmajan solution, this is important here, is directly proportional to the number of viable cells. So, in this way we analyze the cell viability, or you can say the cytotoxicity or the cytocompatibility. So, here we also use some control samples. The control sample means we have to incubate only cell without the biomaterial. So, in that case whatever intensity will come, that we consider as a control, and control means in the absence of any foreign material, and with respect to those growth of cell in the presence of material, whether it is increasing or it is decreasing. So, it is expected it should not be less than 80 to 90%. The cell viability as compared to control, it should be always higher than 80 to 90%. Even sometime if your material is also helping in the cell proliferation, so it may be much higher, it may be 200%, even 300% as compared to control. So, the advantage of this method is MTT can be reduced by different type of cells. So, this method is applied to different cell lines, it may be the fibroblast, osteoblast. So, whatever different type of cell lines you take for a particular application, that MTT can be applied. So, it is relatively inexpensive and it is a reliable method to assess the cell viability, and the procedure is simple and it is quite a reproducible method. But the issue is, in the protocol we have to resolubilize the Formosan crystal or the product. So, that is a tricky part, and especially when there is a low metabolic activity, then we require the higher number of cell to get the desired intensity of the color. So, after in vitro analysis or assessment, we have to see which is the better sample or the best sample, then we go for its in vivo assessment. So, in vivo assessment, they are tested within the living organisms in order to assess, you can say, the actual or the real biocompatibility. So, different models are used, mostly use the animal model, but plant models as well as the even human models can be used for the in vivo assessment. So, in this case, the biomaterials or the foreign materials are administered or implanted within the body, and with time we analyze what is the different interaction with the host tissue or host cell. This analysis gives the idea that what is the response of body to the implant if you are going for its commercialization of that particular implant. So, this is very important. So, whenever a new biomaterial is prepared or synthesized, then we have to go for in vivo assay to confirm its response to the body and its suitability for the full scale applications. So, in case of in vivo assessment, so we can see have the skin irritation test, sensitization test or implantation test. Also we can have the systematic toxicity test. So, here the advantage of this in vivo assessment is we are getting the complete response of body and effect with time which is desired for its actual application, but its demerit includes that after the testing we have to sacrifice that particular animal model. And as compared to the in vitro analysis, this takes longer time. It starts from the it is a few weeks, 3 to 4 weeks, and it goes up to the several months. So, it is a in vivo analysis is a time taking process. So, during the animal studies, there should be the interaction of material with the regulatory, parenchymal, stromatal and the vegetative cell types with implant. Then there should be also the paracrine and the endocrine factors acting on the cell around the implant. So, paracrine means that they regulate the cell cell interaction, and endocrine means they regulate the inside the body or the cellular environmental interactions. Then we just see the cell interaction with the ECM components, and also apart from the ECM components we just see what are the soluble regulatory molecules, because in many cases if you go for implant, so this ECM proteins or these regulatory molecules have been altered by the presence of implant. So, what is the interaction of the cells with the ECM components, or you can say the regulatory molecules due to the presence of implant, and also the interaction with the blood borne cells, proteins and other macromolecules. So, these are the different interactions which take place during the animal study, or you can say in vivo analysis. So, what are the different kind of responses take place during the animal study? So, first there will be a dead space created by the implant, then soluble agent will be released by the implant, like it is some ions or some polymer fragment. So, it depends what is the chemical composition of that particular biomaterial. Also we have the insoluble particle materials, like because of some wear there will be generation of some debris. Then there should be some chemical interactions of biological molecules with the implant surface. So, it includes the surface protein interactions, adhesive protein and cell surface interaction, cell surface interaction. Then also there will be the alteration in the strain distribution of the tissue. This may result because of the mismatch in the mechanical property, or you can say the modulus of elasticity between the implant and the surrounding tissue, and also it may come because due to the moment of the implant relative to the surrounding or the nearby tissues. So, because of some you can say mechanical incompatibility, there will be the alteration in the strain distribution in the tissue, and these factors combine together and give the result, or you can say the holistic response for this in vivo analysis. So, what we analyze during the in vivo assessment? So, we analyze the histology or histochemistry. So, we analyze the relative number of what are different cell types, what is the amount of the ECM components around the implants. We also analyze the presence of lysosomal enzymes, which represents or which indicates about the macrophages. Also we analyze alkaline phosphatases, which indicates about the cell proliferation activity. We also go for the immunohistochemistry. So, we apply this antibodies, let us say monoclonal or polyclonal antibodies to specifically stain a particular molecules. It may be in the membrane, intracellular or extracellular molecules. Then after applying the antibodies, we can analyze using the fluorescence microscopy or some other techniques. Apart from that, we go for the SEM and TEM analysis. So, we analyze that ultra structural analysis, particularly with the TEM. So, we analyze the cells at the interface, and using the SEM we analyze what is we have to fix the cell, then we have to analyze what is the morphology, and also we perform some biochemistry analysis, like particularly the inflammatory mediators. As we discussed, during the inflammatory process all are mediated by different enzymes. What is the presence of those mediators that we can analyze using the biochemistry studies. Apart from that, we also go for the mechanical testing to analyze the mechanical strength or mechanical compatibility. Let us say if you are going for the bone implant, then whether its mechanical property is compatible or comparable or similar with the bones or not. So, these are the in vivo analysis. So, we discuss the in vitro analysis followed by the in vivo analysis. So, whenever we are developing a new biomaterial, so we have to first analyze its physico-chemical property, its surface property, its chemical composition, because surface property is going to regulate the surface protein and cell surface interactions. Chemical property will decide its compatibility, its leaching, how much it is toxic or not. Then cell biological requirement, what is the requirement of the cell, where we are going to apply it, which cell types are particularly for which application you are developing that material. If you are developing for the bone, you are developing for the drug delivery, or you are developing for the heart valve. So, that we have to see what is the requirement. Then its mechanical property, as I told, this mechanical compatibility is very important, otherwise inflammation leading to the replacement of, or you can say the failure of implant. Also we have to see the polymer chemical limitation. So, whatever material you are using, what is its limitation, particularly from its chemistry point of view, what should be the surgical requirement during its implantation. So, nowadays different new surgical procedures are like laser surgery or some other advanced methods have been developed, and also as we know that these complement pathways, they are also activated due to presence of pathogens, especially this lectin pathway or the alternative pathway. So, we have to make sure that before the implantation the material is sterilized. So, you have to find what should be the sterilization procedure without affecting the surface as well as the bulk property of material, to avoid inducing the bacterial contamination or pathogens. So, we have to consider all these while designing or developing a new biomaterials. And finally, a biomaterial is expected to show the better biocompatibility in both in vivo as well as in vitro conditions. So, we start with the in vitro experiment, then we go for the in vivo experiments. So, I will conclude here. So, today we discuss how we define the biocompatibility, and we discuss the biocompatibility is the ability of a material to perform its intended function in harmony with the host response. So, host response will be there, but the biocompatibility criteria will vary depending on a particular application. Then we discuss how we analyze the biocompatibility. So, we started with the in vitro analysis. In vitro we consider its cytotoxicity, hemocompatibility as well as its leachability. These criteria we consider in the in vitro assessment, and for the cytocompatibility we go for the direct method or the agar diffusion or the elution method, and we discuss one example how the MTT assay helps in analyzing the or measuring the cell viability. Then we discuss the in vitro assessment, particularly what happens during the implantation of a foreign material, what are the different cell surface interactions. Then we discuss how we analyze this in vivo results, starting with the histochemistry, histoimmunochemistry, then biochemistry, then SEM and TEM, also the mechanical testing. Then based on this understanding of the biocompatibility, what should be the different aspects that should be considered while developing or designing a new biomaterial. And finally, any biomaterial, it should show the better compatibility in vitro as well as in vivo conditions, which are relevant or appropriate for a particular application. Thank you.